Dilara Hanım orada mısınız?
Evet, buradayım.
Tamam. O zaman yavaştan başlıyorum ben artık. İnsanlar da gelmeye başladı. Okay, hello everyone. Welcome to the first RSG Open Students webinar. This is a new concept that we are trying. We will invite people from the same school and we will give them a day to talk about their presentation, their projects. So it's a nice new event that we are trying to do, and I am so happy that you can be all here today with us. Before starting, we have upcoming events that are not open student seminars. Here are the dates. You can follow us on the social media that I put on the sticky note on the right side of the corner where there's a chat box. Here you can sign up to these events if you want to join them. And if you have any questions, please type them in the chat box on the right side. And if you use the raising hand button, we will see your questions easily because we want to answer all of your questions. Now our first speaker is Dilara Uzuner. She received her BSc degree from Department of Molecular Biology and Genetics at Gebze Technical University. She received her master's degree in bioinformatics and systems biology program at the Department of Bioengineering at Gebze Technical University. Again, she has worked on integrative analysis of transcriptome data and cellular networks to reveal molecular interactions of metastasis mechanisms in cancer research in her master's thesis. Currently, she is a PhD student in Gebze Technical University, and she is working on the metabolic changes in Alzheimer's disease. And without further ado, I would like to give the stage to Dilara Uzuner. Dilara, we can't hear you. Can you unmute your microphone?
Can you hear me now?
Yes.
Okay. Thank you for nice introduction. And I'm also want to start with introduction of our group. We are one of the largest computational biology groups in Turkey at Gebze Technical University. We have master and PhD programs. We have many professors working in different fields of computational biology. And also we have many collaborations with in silico and wet lab groups. Here I share the links of our webpage, our GitHub page, and our group's YouTube page where some course content is shared. You can check our website or university webpage for more information. And today I will present to you our article published from the studies done within the scope of my master thesis. We worked with Professor Devrim Gözeçik and his group on this study. In addition, Assistant Professor Pınar Pir and Associated Professor Tunahan Çakır were my supervisors. And this is the title of our article, Transcriptional Landscape of Cellular Networks Reveal Interactions Driving the Dormancy Mechanism in Cancer. And as we know, cancer is one of the most fatal diseases in the world. Today, many types of cancer can be cured, especially with early diagnosis. However, metastasis remains one of the most important challenges in cancer treatment. Metastasis is the spread of cancer cells from primary tissue to other tissues. And this figure basically explains the metastasis. This is the primary tissue, and some of tumor cells join to blood circulation and circulate along the body. When these cells find a suitable environment, they leave the blood circulation and settle in new tissue. And cancer cells in this new tissue encounter many stresses. For example, because there is not enough vascularization, they cannot reach enough nutrients and oxygen. And the immune cells recognize these cells and try to kill them. There are two paths in front of these cells: dying or finding a way to deal with this stress. And the mechanism that allows cancer cells to survive in metastatic tissue is dormancy. In dormancy, cancer cells are present, but they are not clinically detectable because they are so small in size. And cancer dormancy is divided into two in literature. In tumor mass dormancy, the division rate of cell is equal to the death rate because of the encountered stresses. And for this reason, there is no growth in tumor size. On the other hand, in cellular dormancy, cells enter a quiescent-like state by stopping dividing, but uh, they um, they continue to carry cancer cell properties by regulating their intrinsic mechanisms. Because they are not dividing, they also escape from treatment because chemotherapy or radiotherapy targets the dividing cells. But this is a temporary situation. When cells adapt to their environment, they begin to divide and form tumor cells again, and uh, generally displays a more aggressive behavior than in the primary tissue. In this study, we studied cancer dormancy by subnetwork discovery method. Subnetwork discovery is mapping transcriptome data to whole protein-protein or whole gene regulatory networks to find the most affected parts of the network in the case of interest, the smaller and most affected part of the big network. And in this method, numerical information of transcriptome data combined with the topological information of the interactome data. I will talk about the details of the method later. And I will continue with materials and methods. This is the workflow of our study, and it all starts with transcriptome data. Gene expression measurements of cancer and dormant cells are available in the transcriptome data. First, we perform differentially expressed gene analysis to identify significantly change or not change genes between two conditions. Then we use p-values from here. On the other hand, we also have interactome data. We obtain this data from interactome databases. Then we use two subnetwork discovery tools known in the literature to perform well. And these tools map transcriptome data to the interactome and try to find subnetworks that reach in the significantly altered genes. And in this way, we obtain subnetworks of different transcriptome data. Then we scored the genes found in these subnetworks according to how many subnetworks they were found in. And we created a dormancy interaction network with genes that passed a certain filtering criteria. We used 10 different transcriptome data in this study. All of this data taken from literature, and as you can see here, different types of cancers, different types of experimental methods, or even different types of organisms were used. This is because there is very limited dormancy data in literature. On the other hand, combining all these different studies allowed us to obtain more comprehensive results. And some transcriptome data had more than one compared conditions. That means, for example, for the first data set here, there are two cell lines in the data. So we use these two cell lines in our analysis separately. And as a result, we had 21 dormancy cancer comparison in our analysis. And as I mentioned, we use data obtained by different methods. For this reason, we made differentially expressed gene analysis with according to method appropriate data, and we use Lima for microarray data. We use RNA-seq for DSEQ2 for RNA-seq data, and we use Zimp-Wave algorithm combined with DSEQ2 for the single-cell RNA-seq data. And in this study, we use protein-protein interaction networks and gene regulatory networks. We downloaded these networks directly from the databases, and as you can see here, databases. And in the case of using multiple databases, we combined these databases and unified the data. Also, I want to draw attention to a point here. We use p-values when mapping genes into networks, and algorithms try to get network includes lower p-value genes mostly. However, transcription factors can change the expression levels of target genes without changing their expression levels too much. For this reason, we have also created a TF3 gene regulatory network to not lose information in gene regulatory networks. And you can see the construction of TF3 gene regulatory networks. There is no TFs in the network, but all the genes that TFs interact with are interacting with each other, as you can see here. So we eliminate the TF factor effects, but we protect the gene regulatory information in this network. And then subnetworks were found by mapping each data set to 3 different network types. And as I said before, we use 2 different subnetwork methods, key pathway miner and Bayonet. And the main difference between these methods is the algorithm they use, but I will never go into detail in this presentation of the algorithms. And the last step is interpretation of all subnetworks. First of all, we checked whether dormancy-related genes were found by performing gene set enrichment analysis on all subnetworks. In addition, we checked all genes by comparing them with the relevant Go terms list. In case of there are genes involved in dormancy or dormancy-related mechanism, but not enriched in the first step. We use autophagy, cell cycle, and extracellular matrix Go terms. And also we found dormancy-related genes from literature, and we compare our genes with that list. And finally, we combined all the subnetworks we obtained and scored the genes. When integrating the networks, we use human homologs of the mouse genes, and then we score all genes in the integrated list according to number of appearance in subnetworks, and we had 3 scoring type. First is subnetwork score. Here you can see a small toy example. Here 2 dataset and 3 compared condition. And for gene A, in compared condition 1, it's not found both methods, so its score is 0, network score is 0. And in second compared condition, it's found both methods and the score is 1. And in third compared condition, it's found only by Bayonet, so its score again 1. And the network score is sum of these all compared condition scores. In significant subnetwork score, we also consider the p-value of these genes in the related compared condition. So if its network score is 0, its significance score is again 0. But here, for example, its network score is 1, but its p-value greater than 0.05 in compared condition too, so its significance score is 0. And the significance score is sum of all these scores, all compared condition. And finally, we have also dataset score. Dataset score is the number of appearance a gene in how many datasets. And this is the summary of our work again. There are transcriptome data, genome-wide interaction networks, and we use 2 subnetwork discovery tool, and we found subnetworks, and then we combined these subnetworks to find and to construct dormancy cancer interaction network. And I will continue with the results. Firstly, we compared the power of the tools we use. As you will see here, many of the genes were found with both KPM and KPTV minor and Bionet for the same dataset. But in general, Bionet produced larger subnetworks. So the percentage of genes found only by Bionet is also high. When subnetworks are integrated, we aim to eliminate the effect of the difference between the tools by taking the gene if it's present in a subnetwork, regardless of the method. And we integrated all subnetworks to score genes based on their appearance in subnetworks. We aim to extract the most important genes across subnetworks, and score tables were generated for all networks types separately. And we also filtered the scored genes. Here the filtering criteria: dataset score minimum 2, significance score minimum 3, and a gene should be upregulated in most cases. That means, for example, if gene's significance score is 5, it should be upregulated in dormancy condition at least 3 of the compared conditions. And here you can see the top 10 genes in filtered score tables in all network types. And these are the total lengths of the filtered gene list. And clustering has the highest score in all network types, and it's known that it reduces the sensitivity of prostate cancer cells to chemotherapy and is highly expressed in drug-resistant cancer cells. Also, it has been shown to induce epithelial mesenchymal transition, which is associated with the metastasis mechanism. And his 2H2BE also found commonly in all 3 network types, and it's overrepresented in treatment-resistant breast cancer cells, according to the literature. And FBAX 2022 also has role in epithelial mesenchymal transition. And CDKN2B was reported to attenuate the growth capacity of the tumors and sustain dormancy phenotype. So in filtered score lists, genes in filtered score lists are mainly associated with dormancy phenotype, dormancy-related phenotypes. And then we combined all the genes in filtered score list to construct dormancy interaction network, and this is our network. It includes 139 genes and nearly 2,000 interactions, and all different colored edge shows the different type of interactions. Then we performed gene set enrichment analysis to genes in dormancy interaction networks, and these are the results. And we found many, again, dormancy-related terms. For example, angiogenesis, which is production of new blood vessels to supplement, to reach supplements, and oxygen, response to stress, cell death, regulation of signaling, and p53 signaling pathway, and transcriptional regulation of cell cycle genes, immune system, autophagy, etc. And this is the table of genes that found in dormancy cancer interaction network and uh, previously associated dormancy associated genes that found in dormancy cancer interaction network. And as you can see here, our network contains many genes that have been previously associated with dormancy, and these genes have been associated with different types of cancer and in different models. For example, here colorectal, it is this gene associated in colorectal cancer with dormancy, in glioblastoma, leukemia, etc. And some of them mouse models, some of them human cell models, etc. So, and this provides the comprehensiveness of our network again. Then we performed independent validations. We used two additional dormancy transcriptome data set to compare its results with our network. And we found 7 common genes with top 10 filtered genes with these two independent data sets. And some of them are vascularization related genes and cell cycle related genes. And also we found yeast homologs of our dormancy interaction network genes, and we found that 4 of these genes cause haploproficiency in Saccharomyces cerevisiae. Haploproficiency meaning that deletion of one of the alleles provided growth advantage to yeast cell. Because we choose the overexpressed genes in dormancy when we construct the dormancy networks, this shows that their possible role in suppressing the cell division in yeast. So, in conclusion, we found several genes captured in the network was previously reported linked to the dormancy or metastasis, but majority of the nodes in the network have not been associated with cancer cell dormancy, and the dormancy cancer interaction network provides a molecular framework for dormancy mechanism by combining information repetitively reported in different cancer dormancy datasets from literature, and we hope that targeting the genes featured in this study may increase impact of treatments and prevent recurrence of cancer. Thank you.
Thank you so much for the great presentation. It's very interesting that you are able to create such a web of information that's going to be like really useful in cancer research. Do we have any questions from our audience? I would like to start with a question actually. Uh, let's say, oh, never mind. We have a question from Tulay K. Have you considered the batch effect in datasets as they come from different studies?
Actually, we are aiming to minimize the batch effect with using p-values. Firstly, we found p-values of all datasets with appropriate method, as I said, Seclima or something else, and then we combined these values. So we hope to minimize the batch effect of the studies, and then we get good results. Uh, so we thought we eliminate most of the batch effect, but we didn't do specific thing for that.
I have a question. if there is a new discovery, let's say like there's a new gene that is like suspected that plays a role in a specific cancer, how easy it is that you can integrate the new findings into your database, like the network. Can you do that, or is it like just set in stone and you have to like do a lot of new work to integrate the new findings into the existing network?
Yeah, maybe not just a gene, but maybe if there was a new data set, for example, we can add these data sets results to our network in the same with the same progress first. Find subnetwork, the most important part. Then we integrate the subnetwork to our dormancy interaction network in future maybe.
Okay, thank you. Are there any more questions for Dilara Uzuner? Um, I guess there isn't. If you have more questions, you can still ask, and we will discuss them after the presentations are over. Well, then, I thank you so much, Dilara Uzuner, for presenting your project with us.
Thank you.
Thank you. Now, our second speaker is Ecehan Abdik. She received her bachelor's degree from Department of Bioengineering in Marmara University, and then she received her master's degree from Bioinformatics and System Biology program at Gebze Technical University. In her master thesis, she reconstructed a brain-specific metabolic network of the mouse biohomology-based approach. Currently, she is a PhD student in bioinformatics and systems biology at Gebze Technical University. She is interested in constraint-based modeling, data integration, brain metabolism, and also Parkinson's disease. Welcome, uh,
Thank you
Ejahan.
for this introduction for me.
I'll leave the stage to you now.
Okay, so I can start. I will share my screen. Can you see my presentation? Can you hear the voice?
See your screen.
I can't hear you.
Can you hear me now?
Okay, no, I can hear.
Okay. Okay, we see your screen. You can go.
Okay. Hello everyone. My name is Ecehan and I'm a PhD student at Bioinformatics and Systems Biology program at Gebze Technical University. Today I will talk about our recent article whose title is Systemic Investigation of Mouse Models of Parkinson Disease by Transcriptome Mapping on Brain-Specific Genome Scale Metabolic Network. This study is a result of further studies of my master thesis. And now I also gave my PhD thesis proposal just a few months ago. It will be related to improving biomarker predictions algorithms which combine genome scale metabolic models and transcriptome data integration or omics data integration. And we will try to predict biomarkers for improved algorithm. In my master thesis, I reconstruct the first brain-specific metabolic network model for mice. Then we use this metabolic model to systemic investigation of experimental mouse models of PD at transcriptome level by constraint-based mapping approach, and we predict metabolite level changes between different models. This study was on the cover of the Molecular Omics Journal in August, and it was a surprise for us. Parkinson is the second most common neurodegenerative disease. It's actually a motor function disorders, and patients suffer from motor symptoms like tremor, rigidity, slow movements, etc. And also they suffer from non-motor symptoms like dementia and anxiety. The number of people suffering from PD has dramatically increased in recent decades because of the aged population all around the world. And we don't know the exact mechanism or exact pathology, but the main pathological features of PD are the loss of dopaminergic neurons in substantia nigra region of the brain, and also accumulation of Lewy bodies, which are alpha-synuclein proteins, intraneuronal protein residues. And dopaminergic neurons also produce neuromelanin. So normal and diseased brain have these kind of pigmentation differences, which shown in a part of the picture. And in terms of metabolism, mitochondrial dysfunction, oxidative stress, disorders in redox homeostasis, dopamine metabolism, and also many unknown factors can contribute to neurodegeneration, and these are very interconnected with each other. So metabolism of the Parkinson is very complex and interconnected. In order to investigate such complex interactions and underlying this complex mechanism underlying the disease, system biology can be used by computational analysis and omics data together to understand and interpret such situations. In order to understand disease mechanisms and improve treatments, diagnostic techniques, etc., experimental and clinical studies are mainly carried out on model organisms. Mus musculus mouse is one of the most commonly used model organism for neurodegenerative disease and also other disease, I think. So it's crucial to make a comparative analysis to interpret and translate these studies in mice towards human diagnosis and treatment. And for Parkinson, several experimental mouse models were developed to study disease, and each model presents different aspects of the PD phenotype, and each has some advantages and disadvantages. And mainly there are two main methods to create experimental Parkinson models. First one is chemical toxin-based methods, and second one is genetic-based methods. These are examples of them. For example, MPTP injection causes mitochondrial dysfunctions and loss of dopaminergic neurons to introduce the animal as Parkinson phenotype. And pink-1 gene deletion also results in mitochondrial dysfunctions, and also there are other genetic modifications and chemicals to introduce some Parkinson-related pathological features or phenotypes. In order to interpret and translate these experimental studies in mice towards the human diagnosis and treatment, it's crucial to have deeper knowledge of mouse brain metabolism. For example, there can be some differences between human and mouse metabolism. Genome-scale metabolic networks were proven to be very useful to model metabolic behaviors of pathways, activities, or organisms, and they also allow interpretation of omics data. And establishing the known biochemical reactions in a cell or organism with their corresponding genes and enzymes is called as genome scale model. So if we talk about what is the genome scale model, it's collection of reactions and responsible genes or enzymes mainly. And these collection genome scale metabolic models provide a mathematical platform that enables omics data integration in order to predict condition-specific metabolic behaviors, metabolic activity chains, etc. In this manner, we reconstruct the brain-specific metabolic network for mice at genome scale for the first time by a homology-based approach. We use the available human brain network IMS 570 as a template model. And the reconstruction process was carried by first updating and compartmentalizing the IMS 570, and then addition of new reactions and pathways, and lastly searching for mouse homolog reactions and enzymes. For this, we use many databases like mouse-psych and mouse part of Allen Brain Atlas, etc. As a result, the reconstructed network includes 992 reactions distributed over 48 pathways and 674 genes which control these reactions. And we saw that only 4 of those reactions are different from human version of the model. So these organisms are very similar in terms of metabolisms, only 2 or 3 pathway a little bit different from each other. The reconstructed network then was validated by comparing the flux values predicted by flux balance analysis. I don't know, do you familiar with flux balance analysis or not? It's actually depends on constraint-based prediction. We use some constraints and optimization strategy to predict flux rates by using these genome scale metabolic networks. And as a result, our predictions are compatible with the literature information, such as measured flux rate for mouse brain metabolism, for example, TCA cycle rates, ratio between TCA cycle and glucose consumption rates, or lactate production rates, etc. We compare a few reaction rates. So this model can be used and to predict reliable flux rates. These metabolic networks also use in an integrative manner. For example, integrative usage of constraint-based modeling of genome scale metabolic networks and omics data can be used to examine metabolic mechanisms of diseases. There are many methods which use this integration, and most of them first map gene expression data on models based on the gene-protein reaction associations. If the responsible proteins for the reaction form an enzyme complex, the minimum of their expression value is assigned as the reaction score. If the responsible protein are isoenzymes, which are different enzymes catalyzing the same reactions independently, the summation of their expression values or maximum expression values is assigned as the reaction score. As a result, each reaction has a reaction score representing the state-specific activity level. And I say that there are many algorithms which use this integration strategy. Timber is also one of them. Timber is an algorithm that predicts metabolite biomarkers from the constraint-based analysis of metabolic network. It basically compares the network demands for the production of external metabolites between two states depending on gene expression chains between these two states. In Timber, transcriptome data of control and disease states are separately mapped to obtain condition-specific reaction scores. Then these reaction scores are used to calculate reaction weights that based on reaction score fold chains for each condition. We can see this calculation in left side of the figure. For example, for control condition, the reaction weight is calculated by dividing reaction score of disease condition by reaction scores of control condition. For disease, it's vice versa. Then for each metabolite, before this, we calculate the maximum production rate of released metabolites. So we know which metabolite can be produced with which maximum rates, etc. So then for each metabolite, a flux demand that satisfied high secretion flux, for example, 90% of its maximum rate as a high production used as a constraint. Then we calculate we apply a weight-based optimization strategy, and we calculate the flux demand that satisfy a high secretion flux throughout the metabolite is predicted by minimizing the weighted sum of fluxes. Minimizing weights to the sum of flux means we use these weights as coefficients in our objective function. With this weight-based optimization strategy, reactions with higher objective function coefficients values are forced to take lower flux values. In this way, a flux value or flux vector that's correlated with upregulation, downregulation information at mRNA level is predicted. So if a reaction is downregulated, its reaction rate is predicted lower than the upregulated one. And then network demands are calculated for both conditions for each metabolite. Then network demand differences between two conditions are used to calculate raw production scores. These raw production scores are normalized across the metabolites to obtain timber production score. And high timber score means lower network demand in the disease state and also lower enzyme investment for the high level production of metabolite. And we can interpret this higher tendency for the production and higher potential to being biomarkers because we can produce them with a low investment, low network demands, and more easy way. We apply this algorithm to analyze metabolite level chains of different experimental mouse models of Parkinson disease, and we use 7 different PD experimental Parkinson mouse groups from 5 different transcriptome datasets. These are our transcriptome dataset. Only 2 of them are genetic-based models. The other ones are chemically based. Most of them MPTP injection method. And we analyze the production potentials of 4 T4 secreted metabolites for 7 different conditions, 7 different experimental models. The first and the last 20th percentile of timber score ranking are assumed as potentially changed metabolites. For example, the top 20th considered as potentially increased metabolites, and the last 20th percentile is considered as a potentially decreased metabolites. Metabolites predict to be changed in more than 3 different Parkinson experimental mouse models are considered as potential biomarkers. So we collect the result of all of result from all of the data sets. And we predicted 6 metabolites as a potentially increased in PD: glutamate, lactate, creatine phosphate, neuronal acetylcholine, and formate. Lactate and glutamate are known biomarkers in Parkinson. The produced glutamate is recycled via glutamate-glutamine cycle and TCA cycle in normal brain metabolism. However, in these impairments, impairment of these cycles can cause accumulation of glutamate in synapses, and it has exotoxic effect. And also another information, motor symptoms of PD have been known to be caused by impaired dopamine acetylcholine balance, where the reduced dopaminergic activity in the striatum caused the cholinergic overactivity. About creatine and creatine phosphate, we know that they have important roles in maintaining ATP levels during oxidative stress, which is related to neurodegeneration. And all of these information demonstrates that our predictions are consistent with the literature information. We also predicted 5 metabolites as potentially decrease in Parkinson, which are melatonin, epinephrine, astrocytic formates, and astrocytic bilirubin, etc. We know that dopaminergic neuronals, which is the main pathologic behavior of Parkinson, caused a decrease in catecholamine levels like dopamine, epinephrine, adrenaline, etc. And this confirms the predicted decrease of melatonin and epinephrine in most of the PD mouse models. Our predictions show that most of the models behave like melatonin and epinephrine levels can be decreased. However, dopamine decrease was only predicted for 2 Parkinson models, which are pink-1 knockout mouse model and MPTP early mouse model. This early means the sample taken from sample taken after the short time after the MPTP injection. The late versions, in late versions, samples are taken after a few days and maybe more. So the other well-known dopamine level related pathology of Parkinson, neuromelanin loss. This is also this was also predicted by these 2 models. Then we cluster mouse model samples based on the calculated timber scores, and we saw that these 2 experimental models clustered together. Both models, pink-1 knockout and MPTP injection, create PT pathology by causing mitochondrial dysfunctions. However, other MPTP-based Parkinson models could not reflect the PD metabolism as good as MPTP early group. The main cause of these differences can be attributed to the model creation methods, because genetic modification can have lasting effects on metabolism, whereas chemical treatments effects may not last permanently. According to Timber results, more accurate biomarker predictions were made for MPTP early group, for which the samples were taken 24 hours after injection, compared to other MPTP groups, for which the samples were taken 4 or 7 days after injection. As a result or conclusion, these reconstructed brain-specific genome-scale metabolic network and its integrative use with transcriptome data provide a comprehensive investigation of metabolic changes in the experimental mouse models, and we saw that none of the experimental models can reflect the PD metabolism exactly at metabolic and transcriptome level. So experimental animal models still need to be improved, and these kind of studies, computational studies, can help these improvements. We can show a way, and this approach can be applied for other neurodegenerative disease to improve animal models in feature studies, and also maybe for other kind of disease with different genome scale metabolic networks. My ongoing studies are generally related to improve biomarker prediction algorithms and also predict biomarker for PD by using main datasets. We try to collect all available omics datasets related to this disease. And I think that's all for me. Thank you very much for your attention. These are our group members in Gebze Technical University.
Okay, thank you so much for the great presentation. It's very nice that you are trying to combine the two different mouse models, the chemical and the genetically modified ones, to try to get answers for Parkinson's, I guess. Someone asked if they could ask a question. Yes, you can ask. Just type your question in the chat box and it will be asked. I have a question. The scoring system. So you said it has to be changed in 3 different models, the metabolites, to be considered as a potential biomarker. Is there a reason why you picked 3?
We tried to at least more than 3, because we thought that it should be seen in more than half of the data sets to make the study more reliable, because some of the models cannot reflect the disease mechanism. So we try to make more reliable results. So we combine many data sets, and generally we consider the results which are same for more than half of the data sets.
Okay, thank you. Do we have any other questions for the presenter? So Yaren, you can still ask your question. We can ask it after everything ends. Now we will take a 5-minute break, and after that we will continue with Hatice Büşra Dülceli. And thank you again for presenting.
Thank you.
Welcome back, everyone. Next, we are going to continue with Hatice Büşra Lülceli. She received her BSc degree from Department of Bioengineering at Yıldız Technical University in Istanbul, and then she received her master's degree from the same department. Her master's thesis was about the analysis of intratumoral heterogeneity in context of tissue-specific gene expression by using a new computational approach. Currently, she is a PhD student in bioinformatics and system biology program at Gebze Technical University. She is interested in constraint-based modeling, data integration, brain metabolism, and Alzheimer's disease. If you are here, Hatice, you can start.
Yes, I'm here. Thank you for your very good introduction. Okay, I'm sharing my screen. Okay, can you see my screen? Do you hear me?
We hear you and we see your screen.
Is it okay?
Yeah.
Okay, thank you so much. Okay, hello everybody. Actually, I will talk about a part of my PhD study, and the title is Imet application as an integration method in Alzheimer's disease in order to predict reaction activity. And I'm sorry for waiting. Yeah. Actually, I will explain some information about Alzheimer's disease and the alteration of cholesterol metabolism. It's very important metabolism. It's in fact that affected during the progression of the Alzheimer's disease, and I will explain the whole of the study. And we are focusing on Imet models, and I will explain results. So, the Alzheimer's disease is the most common cause of dementia in all over the world, and around 130 million people will be affected in Alzheimer's disease in the future. It's a very important number for us. And loss of cognitive functions and memory is very important characteristics of Alzheimer's disease. And we know that there are many biochemical processes, metabolic pathways are affected in the Alzheimer's disease. So actually, the brain regions are affected during this progression of this disease differently, and we know that from the literature, specifically internal cortex and hippocampus are more affected brain regions in these diseases. And there are some affected metabolic pathways and molecular chains in Alzheimer's disease. First one is we know that amyloid beta and tau protein accumulation from between cells and among all neuronal cells in the brain. And oxidative stress and mitochondrial dysfunctions are very important processes in Alzheimer's disease. After these dysfunctions, cellular energetics are altered, and very important dysfunction in here because of mitochondrial dysfunction. And then there are other pathways related to neurotransmitters, lipids, cholesterol, sphingolipids, and bile acids processes. And finally, neuronal death is very important in the progression of Alzheimer's disease. So we can say that there are lots of affected metabolic pathways in this neurodegenerative disease. So we focus on cholesterol metabolism in Alzheimer's disease, and we know that accumulation of beta amyloid protein is related to alteration of cholesterol metabolism, cholesterol biosynthesis, and also cholesterol catabolism. Unfortunately, the exact impact and mechanism of cholesterol metabolism are not clear. So we focus on the alteration of cholesterol metabolism in our study. This is our study actually. There are many other points in that study, but I will explain the Imet models and Imet analysis based on metabolic models. So the main purpose of this study is to detect the role of brain cholesterol metabolism in Alzheimer's disease. This metabolism includes the biosynthesis of cholesterol and catabolism of cholesterol. After cholesterol catabolism, we obtain bile acids, and alteration of bile acids in Alzheimer's disease is very important because cholesterol and bile acids are very important for the homeostasis of the brain in human body. The other purposes of this study is to test metabolite markers of brain cholesterol biosynthesis and catabolism. We used two brain autopsy samples and for two different brain regions, like that, inferior temporal gyrus and middle temporal gyrus. The other important point is to detect difference in gene expression level between Alzheimer's disease and healthy people using ANOVA test. Actually, my focus in this study is here. It's that genome scale metabolic network modeling to assess alterations in cholesterol biosynthesis and catabolism reactions in Alzheimer's disease for three different brain regions: hippocampus, internal cortex, and primary visual cortex. Finally, we tested the Parkinson's disease with the same analysis to understand these alterations are specific to Alzheimer's disease or not. So I will explain Imet algorithm. What is Imet? Imet is integrative metabolic analysis tool, and actually Ecehan said lots of things about the mapping of gene expression data to metabolic model. The steps are generally same, but there are specific things in different algorithms, and I am focusing on Imet algorithm. It's an optimization method, and after Imet algorithm, we obtain condition-specific models. So we have a gene expression data for healthy people and patients with Alzheimer's disease, and also mild cognitive impairment patients. We have gene expression data, and we have a metabolic model. There are lots of different reactions, and there are lots of different pathways in this model. After mapping gene expression to this model, we obtain like that. We obtain this condition-specific metabolic model. So Imet shows us activity of the reactions in that model based on our condition. So we can say something about alteration of metabolic chains in Alzheimer's disease or any other disease, what you want. So we use human GAM. It's a large human metabolic model. We use this metabolic model for a metabolic model, and there are lots of reactions, metabolites, and genes in that model. Gene information is very important because we use this gene information for mapping process, and we have two different microarray data sets from GEO, and there are information, gene expression level information for internal cortex, hippocampus, posterior cingulate, and primary visual cortex. We performed all analysis for four for all brain regions, but we combine entorhinal cortex and hippocampus data after detecting their specific metabolic models. And in our study, we compared the entorhinal cortex, hippocampus, and primary visual cortex based metabolic change in Alzheimer's disease. So how Imet algorithm works? The figure is summarized the Imet algorithm. And again, we have a transcriptome data, we have a metabolic model, and we know that the gene expression level of all genes in that data. And we have two thresholds, we detect two thresholds. And there is a small basic metabolic pathway here, and we know that genes or enzymes controls reactions. So according to this Imet algorithm, if a gene or an enzyme has lowly expression, it is predicted as to be post-transcriptionally down-regulated based on that, because gene 1 and gene 2 has low expression, gene 3 has high expression. Based on these two, this reaction 5 is predicted as inactive reactions or post-transcriptionally down-regulated reactions. The other example here in the paths, there are two genes or two enzymes. They have high expression, and here gene reaction or gene enzyme reaction is controlling reaction 6. And this gene 6 has moderate expression based on our thresholds, two thresholds. And according to Imet algorithm, this reaction is predicted to be post-transcriptionally upregulated because of these two genes, because they have high expression, and this reaction is predicted is active reaction. So after prediction based on Imet algorithm, we have specific metabolic models based on our condition. We performed Imet algorithm for control samples or healthy people and also Alzheimer's disease people. And after detecting activity of these reactions in the model, we binarize this information. What is binarization in that condition? If a reaction is active, here, for example, reaction 1 is active, we can label this reaction as 1. And then, for example, here, reaction 5 is inactive, and we can say that this reaction is 0. Okay, after, based on this binarization, we performed Fisher-Exact test to understand statistically significantly difference or significantly alteration in reactions during Alzheimer's disease. Another important point here, this algorithm or this simulation was performed for all samples separately for control and Alzheimer's disease. So we obtain, after Imet simulations, we obtain context-specific or personalized metabolic models for each people. And there is another important point for Imet algorithm. We know that generally in mapping algorithm from gene expression or proteome data to metabolic models, we have measurement constraint specific to our condition. However, in Imet algorithm, this is an advantage for Imet. We don't have measurement concept. We don't use measurement concept, but we use three of reactions. They are very important for living brain. If a brain is living, glucose and oxygen and biomass reactions are very important and we need them. So we use just 3 of them for a measurement constraint, but there is no any other specific measurement constraint in that algorithm. It's very important and it's very important advantage for Imet algorithms. So we use 2 different data sets from Geo, and before using this for Imet, we performed age and sex correction for these 2 data sets. And there are different number of samples for internal cortex and hippocampus, and the other data sets like that. And we combined the internal cortex and hippocampus specific Imet models after binarization of the data. And so it's a small figure for all I explained before. We have a huge human model. After application of Imet, we have specific context-specific models for each healthy people and also each Alzheimer's disease patient. And after Imet, we have new metabolic models for each people, and these models has number of reactions like that. I want to remind you, human gen has around 30,000 reactions and 3,600 genes. After Imet algorithm, after mapping of expression data to model, we obtain these number of reactions and genes in specific models for healthy and Alzheimer's disease patients. So after a Fisher-Exact test, we detect significantly changed reactions in different brain regions for both 2 data sets. There are lots of reactions and lots of different metabolic pathways that are affected in Alzheimer's disease. For example, sphingolipids, glycosphingolipids, lots of different amino acids, cholesterol, bile acids, lipids, fatty acid metabolisms are affected in progression of Alzheimer's disease. So these are the number of reactions that are affected, that are altered in Alzheimer's disease in the different brain regions based on data sets. So actually, we can say these results, based on these results, internal cortex and hippocampus are more affected compared to especially primary visual cortex as a difference in brain regions. So this is the summary of cholesterol metabolism. It's from the study, from the article. These 2 paths shows us the cholesterol biosynthesis. There are 2 different pathways, presqualene pathway and postqualene pathway for cholesterol biosynthesis. After cholesterol biosynthesis, these 2 of them show us cholesterol catabolism. So after catabolism of cholesterol, we obtain bile acids from cholesterol. This is enzymatic and this is non-enzymatic. We focus on enzymatic cholesterol catabolism because we have enzyme information for that. So we have gene expression level information. So we have information for enzymatic cholesterol catabolism. And then this figure shows us the difference of control and Alzheimer's disease patients based on gene expression level, just gene expression level. So green boxes shows us the lower lower metabolite concentration or gene expression level. The red one indicate the higher metabolite concentration, and the gray one, there is no significant change in that enzymes or genes or these reactions. So we can say that some of them are affected in Alzheimer's disease just based on gene expression level analysis using ANOVA. But and then we can say that a more pronounced down-regulated reactions was specifically predicted in pre-sequelae pathway for hippocampus brain region. And post-sequelae pathway was more significantly affected in internal cortex. So they are different from each other. And several reactions that are affected in Alzheimer's disease are related to cholesterol catabolism, such as 26-hydroxycholesterol metabolism. Hydroxycholesterols are very important. They are the pre-metabolite of bile acids, and they control the brain homeostasis, and so their alterations is very important to understand brain cholesterol and bile acid metabolism. Finally, we can say that vitamin D production is another important affected metabolite during Alzheimer's disease progression. And this includes the reaction information for cholesterol catabolism and also biosynthesis, and some of reactions are affected again. We can see again some of them are affected significantly for internal cortex and hippocampus, but there are just one reaction is affected in primary visual cortex. So we can say that in our analysis, we demonstrate that internal cortex and hippocampus are more affected brain regions during Alzheimer's disease progression compared to other brain regions. Okay, I want to remind this figure from here to here, and there are boxes, green boxes, and this information was obtained based on gene expression analysis, but we performed Imet analysis. We obtained Imet models specific to healthy and Alzheimer's disease patients, and after binarization of activity of the reactions, we performed a statistical test, and then we can say that these reactions are statistically significantly affected in Alzheimer's disease. So these green boxes support the gene expression analysis for these reactions, like that. These are these gray boxes on the other here. These reactions are there are no significant changes based on gene expression level. However, after adding metabolic analysis via Imet model prediction, we can say that these reactions are also affected in Alzheimer's disease progression. And here there is a very important point, because this shows us bile acid metabolism also affected during Alzheimer's disease progression, and this information was detected based on Imet models. And then actually the study is like that, and we want to understand these metabolic chains for cholesterol metabolism and bile acid metabolism are specific to Alzheimer's disease or not. For these purposes, we performed all same analysis for Parkinson's disease using two different microarray data set, and then we can say
Oh, okay. You're in conclusion. I was gonna say
Yeah. The
Okay.
last
Sorry.
slide. Okay. We can say that Alzheimer's disease is associated with abnormalities in cholesterol biosynthesis and catabolism, and different brain regions are affected differently from cholesterol biosynthesis, pre-squalene or post-squalene. And these changes, these alterations are specific to Alzheimer's disease because they were not detected in Parkinson's disease, and abnormalities in cholesterol metabolisms are possible therapeutic targets in Alzheimer's disease. We can say that. Okay, thank you so much. I will just say something. My advisor is Dr. Tuna Çakır, and we are collaborated with Vijay, Anup, and Madhav from National Institution on Aging from the USA. We are the same research group with Dilara, Ecehan, and Müberra. If you have any question in later, you can send email. Thank you so much.
Okay, thank you so much for the great presentation. It's very interesting that cholesterol is such a, I guess, unique part of Alzheimer's disease, since you don't see it in Parkinson's. There are two questions for you.
Okay.
First one is there any specific reason for selecting Imet? Imet was developed back in 2007, and I believe that more robust algorithms have been developed since then.
Actually, we use human gem. This is a last version of human metabolic model. It's very huge model. It includes lots of reactions, thousands of reactions. And we have no information about brain metabolism in that model. The advantage of Imet algorithm, we don't use any measurement constraint. We don't need any measurement constraints. So we can use this if there is no information about the situation. We can use Imet. For this reason, we select Imet algorithm.
Okay, and the second question was if this was a modified version of Imet, but I guess you also answered that question. Other than that, do we have any other questions for Hatice? I guess there is no other question. In that case, I have a small question, but I'm not sure if it's worth asking. But can you use Imet to later, can you use Imet to link the abnormalities in cholesterol pathways with symptoms of Alzheimer? Can you, for example, say this happens and because of that, dementia, and this other pathway leads to motor skill abnormalities, etc.
Actually, after Imet analysis, we obtain specific models to patients or to healthy people. And for example, human gem has 13,000 reactions. After mapping via Imet, we have a model specific to a patient, and it includes, for example, 7,000 reactions. And these reactions are coming from different pathways. So we have information, the alteration of other pathways. Just we focus on cholesterol metabolism. So we have the other pathways activity after Imet. Is it clear?
Yeah, I understand. Well, Imet's pretty interesting. It's nice tool, it seems. Okay, then let's continue with our last speaker now. Thank you so much, Hatice, for your presentation.
You're welcome.
Here, real quickly. And our last speaker for today is Müberra Fatma Cesur. She received her bachelor's degree from the Department of Molecular Biology and Genetics in Gebze Technical University, and then she received her master's degrees from Bioinformatics and System Biology program under Bioengineering Department, in addition to the Department of Molecular Biology and Genetics from Gebze Technical University. In the scope of her thesis, she investigated the effect of the induced stringent response of antibiotic production from Streptomyces coli through in vitro approaches. Furthermore, she worked on identification of the putative drug targets in Klebsiella Pneumonia through in silico gene and metabolite-centric approaches. Currently, she is a PhD candidate and a research assistant in the Department of Bioengineering in Gebze Technical University. She keeps her studies on metabolism-centered systems biology under the supervision of Associate Professor Dr. Tunan Çakır. And with that, the stage is yours, Mubera.
Thanks so much for the nice introduction. I'm sharing my screen. Can you see my screen? I shared my presentation. Yeah. Okay. Now I'm a PhD candidate in the Bioinformatics and Systems Biology department, and I will talk about my master thesis related to identification of putative drug targets for Klebsiella pneumonia using genome metabolic network modeling approach. And our study was published in Frontiers in Cellular and Infection Microbiology journal. Firstly, Klebsiella pneumonia actually is a member of Enterobacteriaceae family, and this pathogen was first observed in the patients suffering from the pneumonia. This gram-negative facultative anaerobic bacterium can survive diverse conditions like human body or surface of the several medical devices, soil, and so on. Klebsiella pneumonia actually is a prominent pathogen because it's a member of escape pathogens. Escape means the ability from escaping from the antimicrobial molecules via development of the resistance. So these pathogens can develop resistance, the antibiotic resistance against the antimicrobials easily. And hypervirulent Klebsiella strains can cause serious diseases like pneumonia, meningitis, and so on. Considering the increasing antibiotic resistance, spread of the resistant, multi-drug-resistant Klebsiella strains, and the lack of efficient therapeutic approach, we focused on the metabolism of this pathogen. Using genome metabolic network model, we try to understand the metabolism, comprehensively understand its metabolism, and also identify putative drug targets. Actually, genome metabolic network models are mathematical representation of whole metabolism, including all the reactions, metabolites, and genes within the organism or cell. And we used IBL model of the Klebsiella pneumonia. Firstly, we created the biomass equation in the model by adding the universally essential cofactors of the prokaryotes into the biomass equation. Then we performed all simulations under the host mimicking conditions, including human body fluid and sputum macrophage condition. Actually, sputum means the infected accumulation of different compounds in the infected lungs. And we know that Klebsiella pneumonia can survive in the alveolar macrophage. So we combined both conditions to design a restricted growth condition for this organism, Klebsiella Pneumonia. And then we searched putative drug targets using two different approaches, including gene-centric approach and metabolite-centric approach. In the first part, I will explain the gene-centric approach. We firstly identified essential genes using gene essentiality analysis, and then we determined the non-homologous essential genes to avoid any potential side effects in the human. And these genes, these non-homologous essential genes, were called as drug targets. And then we characterized these genes based on several criteria like localization, drugability, virulence, and broad spectrum. And we used several databases and literature to identify these factors, these characteristic features of the pathogen. As I said, firstly, we identified non-homologous essential genes for the minimal side effects. And then we prioritized these targets to identify the most effective targets. This figure shows the number of essential genes, number of putative targets during the filtering steps under two different growth conditions. Firstly, we performed gene essentiality analysis. Then we identified non-homologous genes. And then we identified druggable genes, virulence factors. And lastly, we analyzed the conservation of the genes in the Klebsiella Pneumonia. And the genes that are non-homologous and essential were called putative targets, and we firstly characterized these targets based on the NH pathways. And we identified actually several pathways that are prominent for this organism, like membrane structure, amino acid biosynthesis, cofactor production, lipid metabolism, and so on. And also some pathways were determined as specific to the SM condition, like pentonate pathway or fatty acid metabolism. And then we characterize these putative targets based on the localization information. Localization is important to determine the characteristics and function of the proteins. And also we know that outer membrane proteins and extracellular proteins are vaccine targets. But cytoplasmic and periplasmic proteins, if we want to target cytoplasmic and periplasmic proteins, we should design small drug molecules to provide the passing the membrane structure. And our targets are predominantly the cytosolic proteins, and several genes are related to inner membrane. After identification and characterization of the putative drug targets, prioritization of these targets was performed to identify the most efficient targets, because we have a huge solution space, we have a huge target space. We should narrow down these targets. Firstly, we identified druggable targets for higher drug susceptibility, and then we determined the virulence factors because we know that the virulence characteristics is related to pathogenicity and drug resistance. And finally, we focus on the broad spectrum targets. Broad spectrum means the targets can be found in different pathogenic strains, and these conserved targets are important to delay the drug resistance developments, and we can target these proteins to prevent the co-infections and multiple infections as well. So we explored several genes. These genes actually are involved in different metabolic processes related to cell membrane. Actually, the membrane of the gram-negative bacteria includes capsular polysaccharides and lipopolysaccharides. These structures are significant to protect the organism from antimicrobial compounds. Lipopolysaccharides are also important for the stability of the membrane structure. Actually, based on the criteria, as I said here, we identified KDCA protein is the highest ranked drug target, so we focused on this target. Actually, this protein is related to synthesis of the KDO molecule. KDO provides to link lipid A and lipopolysaccharide molecules. And actually, KDO is important to link these essential molecules because lipid A molecule is also essential for the survival of the pathogens and membrane stability. And inhibition of the KDO synthesis resulted in blocked replication and cell growth arrest. And we also found that there are different studies in the literature related to development of the KDO inhibitors for different pathogens. So we focused on this protein, and using structure-based virtual screening, we actually identified two potential KDCA inhibitors. And in the second part, we focused on the metabolite-centric approach. This approach is similar to gene-centric approach. Firstly, we identified essential metabolites based on metabolite essentiality analysis. And then we eliminated the metabolites that are also found in the human metabolism to avoid any side effects in the human as well. And this process also eliminates the currency metabolites. Currency metabolites that are general metabolites found in many different reactions like ATP, NAD, water, and so on. So we eliminated these metabolites. And then we also removed the metabolites involved in the reactions catalyzed by human homologous enzymes. We determined the essential metabolites. This metabolic-centric approach is especially important to identify the essential metabolites because the structure of the metabolites are analogous to the drug structures. So if we identify any essential metabolites, important metabolites, we can design drugs similar to its structure, and these drugs can simultaneously inhibit multiple outgoing reactions, and also we can target synthetic little interactions, and it is important for more efficient treatment. And then we determined the genes related to these essential reactions. These genes were called putative drug targets. Using this approach, we detected several potential drug targets related to riboflavin synthesis, peptidoglycan synthesis, fatty acid synthesis, and so on. And these putative targets are related to these essential metabolites. And we can design drugs that are structurally similar to these metabolites to inhibit these targets. So these drug molecules can inhibit, for instance, this drug molecule, a drug molecule that is similar to these metabolites, can inhibit different genes in the same time. related to peptidoglycan synthesis. So this approach is important for simultaneously inhibition of different targets. These targets are associated with peptidoglycan synthesis, and inhibition of the peptidoglycan synthesis can cause cell lysis and death. And another target, FABI protein, is involved in the fatty acid synthesis in the membrane structure. And actually, there is a significant effort to develop inhibitors against this protein. And also, we identified some genes related to riboflavin synthesis. Riboflavin is important for the synthesis of FAD molecules. As you know, FAD is a general cofactor, and it is essential for many different enzymatic reactions. So all targets can be evaluated much more in future. As a summary, we performed two different approaches to identify putative drug targets in Klebsiella pneumonia. And to our knowledge, this is the first attempt to determine putative drug targets for Klebsiella pneumonia in a such comprehensive approach, using such comprehensive approach. And we identified a putative inhibitor in this study. It is a derivative of coumarin with low toxicity and high antimicrobial potential in mice. And we know that there are different coumarin-based compounds that are efficient in the inhibition of Klebsiella pneumonia. So this inhibitor molecule can be investigated much more. And then we also suggested several drug molecules based on the scoring approach. But there are many other genes, many other essential and non-homologous genes. And these genes can be also further analyzed and characterized. And our targets are mainly related to cofactor metabolism and biosynthesis of cell wall. They are growth-related processes, and we know that suppression of growth-related processes can cause development of the resistance rapidly. So we can also focus on another process like virulence. We can identify virulence-linked targets using another approach. For instance, we can optimize the growth-dependent virulence factor synthesis, and we identified the virulence important virulence genes. And this approach may provide identification of the virulence factor by delaying the antibiotic antimicrobial resistance development. Thank you for your attention, and also I would like to thank you, Dr. Tunan Çakır and Salih Durmuş, for their supervision. And also I would like to thank Riyazuddin and Büşra Şiraz for their contributions to this thesis. Dr. Riyazuddin and Büşra Şiraz performed the structural analysis performed in the thesis. And so that's all. Thank you.
Thank you so much, Berra. Do we have any questions from the audience? I have a more general question about pneumonia. I don't know how to say it,
Okay.
but it's from what I know, and it's like from what people usually say, it usually like follows like an influenza infection, because personally I have never heard of someone directly developing pneumonia like before they had some kind of upper respiratory infection. Is this actually true, or can you just get pneumonia before getting any kind of respiratory infection?
Actually, Klebsiella pneumonia also affect the lungs, and they can cause some respiratory problems. But especially multi-drug resistant Klebsiella pneumonia can cause serious effects. But normally we have Klebsiella pneumonia in our microbial flora as well. But they are not dangerous when compared to multi-drug resistant strains. They can be only dangerous to the person, immunocompromised person. But multi-drug resistant strains are dangerous, like influenza or other. This is actually they can cause serious illnesses.
So I guess the other diseases also lower your immunity, and then you become very susceptible to actual danger.
Yes, actually, co-infections can increase the danger of the microbial infections, including the Klebsiella infections, because these diseases can affect your immune systems. And so co-infections are more dangerous. And you can get actually Klebsiella and other pathogens can cause the co-infections, and this situation is more dangerous. Yes.
Okay, thank you so much.
Thank you.
Do we have any questions? If I guess not, in that case, I would like to thank all of our speakers and all of the people that attended today. This was our first open student seminar, and there will be more. If you want to do a presentation, you can check our website, and there's a sign up form. And with that, I'm ending the seminar. Thank you again, everyone, for joining, and I hope you have a nice day.